particularly pleased to see a really good number attending. I've asked that anyone who's got their cell phones turned on can turn them off. To introduce myself very briefly, I'm Fiona Coma. I'm the owner of a Hoof Wall affected pony who's been part of this research. I didn't know I was going to be chairing this until about 5 minutes ago. So, I'd like to introduce, I'll talk briefly about how the afternoon will run. Carrie Finno will speak. We'll have questions after that. Then there'll be a short break for afternoon tea. Following that, Ray Knightley will talk. He's a farrier with experience and dealing with several ponies with this condition. We'll have some questions for Ray, and then Sheila Ramsey will talk about some implications for breeders and people who are involved with the breed. We'll have a few questions for that, and then we will wrap it up. So firstly, I'd like to introduce Dr. Carrie Finno. She did her veterinarian's degree at the University of California, Davis. She did her PhD in genetics in 2012. And she's now Assistant Professor of Veterinary Genetics at the University of California, Davis. Carrie.
Thank you for the introduction. Thank you all for coming this afternoon, and thanks for the invitation and support in getting me to Ireland. But to actually really talk about this very important disease that I think a lot of people in this room are incredibly passionate about. In fact, I know they are, and that due to their efforts and working with us at UC Davis, we've actually been able to do a complete genetic investigation and very recently uncover the genetic mutation. So this is a huge effort on the part of many people. And I do want to acknowledge the co-authors on this study. So Dr. Danica Banish, head geneticist at UC Davis. Carly Stevens was a vet student working on the project. Amy Young, our technician. Dr. Verena Offalter is a pathologist. So she looked at a lot of the sections of bugs. And then Sheila Ramsey, who's really spearheaded this entire project. So many of you have been walking around looking at the posters and seeing some of the stories about these affected ponies. So typically clinical signs of this disease develop during the first year of life. So most owners report abnormalities noticed during kind of 1 to 6 months of age. All 4 feet are affected, and then in kind of talking to Jocelyn, sometimes the hind feet might be more severely affected. And the ones we've seen, typically it's all 4 feet. And there's cracking and peeling along the hoof wall. And so what you'll see in these affected ponies is this kind of retraction here of the dorsal hoof wall. And it'll look really brittle and cracked on the solar aspect. And so horses, ponies, everyone, walks on their dorsal hoof wall, right? That part of the hoof is what actually sustains weight. They don't walk on the sole, they walk on their dorsal hoof wall. And so if as they're walking on this is actually cracking back, they end up having to walk on their sole. And what you end up getting is this proliferative sole, and I'll show you pictures of that. But this is more of the dorsal hoof wall here that is cracked off. Sheila's kind of said that sometimes people will refer to this as a coconut matting appearance, as you see it crack back. And then this is what happens. So the pony, because now they have no weight-bearing surface, the sole, or the bottom of the hoof, will proliferate. And they'll get this incredibly thick callus, to the point that in this case, I mean, you'd be hard-pressed to identify the frog, right? It's there, but it's pretty much surrounded by proliferative sole. And so they can stay reasonably comfortable with all of that sole, but as a farrier, if you go and start to trim that sole down, they'll often get sore again, because that's not the part of the hoof they're supposed to walk on. So a couple more pictures of some severely affected cases, and then when you look along the solar margin, even after trimming, you can often still see that fissure. So this is kind of what we've seen clinically. And then a very severely affected case where now the dorsal hoof wall is retracted about halfway up the hoof. So we were contacted initially at UC Davis and asked to investigate this. And you know, there's always a question, is it genetic, is it environmental? And as all of you know, Connemars are very well known for having excellent feet. That's really what they're bred for. So the fact that we had an early age of onset, Sheila had really looked at a lot of pedigrees and shown that there were some connections. The fact that parents always appear normal for the most part, and that foals are affected, kind of led me to think this may be genetic and it may be autosomal recessive. And then we actually had this case. I had a veterinarian who's a student of mine that graduated a few years ago, and he works now in Sonoma. And he called me and said, I was out looking at a 6-month-old Connemara filly, and she's got this really strange cracking of her hooves. And I told the owner it's probably white line disease. Let's just do some, treatment. And the owner said, no, I've been on the internet, and I think it's this hoof wall separation disease, and I think it's genetic. And so Taylor, the veterinarian, called me and said, you're the only one I know that talks about genetics. Is this genetic? And I said, actually, we're working on that. Can you send the filly in so we can look at her? So this is Megan Lepper's filly, who many of you may know, and she was nice enough to be happy to let me share these videos of her and Mrs. Crafty. So this filly came in, this was actually on a Saturday at UC Davis. We've got other trailers coming in, and she was pretty comfortable on this ground. She's a little footsore. When we went to trot her, she was actually quite lame up front. And she's the one I showed you that had the pictures of the very proliferative sole. So her feet had cracked back so badly that she had this proliferative sole. And importantly, here's her dam standing behind her, who was barefoot and had very normal feet and was really comfortable. So this was just Megan kind of picking her foot up so that we could see the surface of it. And this is where I got those pictures from. So you can appreciate as you kind of run your finger along, you can actually get into that crack where the dorsal hoof wall there has broken away. And then all of that extra tissue, that's all solar proliferation. So that's the sole that's just trying to make it callous in order to try to keep her comfortable. So as we brought them in, they allowed us to obtain DNA samples from them. And then the other thing I did with her, and actually with 2 other affected ponies that were both in California, was to do a complete physical exam. So in humans, there are a lot of diseases that cause skin abnormalities, but they often also cause mucocutaneous problems. So the gums will be affected, the teeth may be affected, lots of connective tissue problems. So as a geneticist, I look at it from a comparative aspect. So we took x-rays of teeth on all of these ponies. They were completely normal. We cannot find lesions anywhere else. Hair coat appears healthy, skin appears healthy. It is strictly confined to the foot. And this is incredibly unique. Even in cattle, you don't see any genetic diseases that are only confined to the foot. Coronary band looks normal. Often they're lame at the walk. And then in the early stages, if you take x-rays, you don't see any abnormalities. The alignment is perfect. But as they continue to walk on these hooves, make that proliferative sole, get more and more sore, there's inflammation and they often end up foundering. And so we see them later on in life where they develop laminitis. So just some histologic pictures of what this looks like. We had some owners that were very generous in that when they did make the decision to put their affected ponies down because they couldn't maintain their quality of life, they contacted me and said, we want to donate their hooves and anything else you need. And so they sent these hooves to us and we were able to do a complete pathologic examination on 3 cases. So there's, again, this dorsal hoof wall separation. You can see it in these beautiful plasticized figures here too. Occurs in the dorsal hoof wall. The white line is intact. The white line is completely normal, both grossly and histologically. So under the microscope, it's normal. The lamina are also normal if they haven't progressed to a stage where they're foundering. And the coronary band was normal. So it's really confined to that dorsal hoof wall region, which was, again, a very unique situation. So owners that have these ponies, many of them will keep them trimmed every 6 weeks. A lot of the farriers that I've spoken to, and I'm a geneticist, I'm not a farrier, so I do my best to kind of understand how they maintain them, but they say the worst thing you can do is actually take all of that sole off. That sole is protecting them and keeping them comfortable. So they try to just kind of trim away some of that broken hoof wall, but leave the sole alone. And this is a pony before and then after trimming. And you can improve the appearance. You can make it look pretty good, but it is something you really have to keep up on. Some of them will remain sore regardless. People have tried many different types of shoes. Often nails won't hold. And I've seen lots of hooves where the nails will actually be a source for the dorsal hoof wall to crack. People have tried glue-on shoes and some other types of shoes. So they do require some pretty intensive treatment. So then back to the genetics, there were common bloodlines among affected foals. However, the parents appeared normal. And so this, to me, suggests an autosomal recessive mode of inheritance. And we're just going to go over really brief genetics in that, in this case, if you have the dam, who we had in Megan's filly's case, she was completely normal hooves, bred to a stallion. This isn't the sire of this filly. This is a random gray stallion off the internet. So those of you who know them all and what they look like, I know it's not the sire. And then this is his hoof here. There's a 25% chance that you're going to produce an affected foal. And the reason for this is that you've got a copy, so you've got carriers. The dam is a carrier, the sire is a carrier, and we'll go through a Punnett square kind of at the end. They each have a copy of the gene, so there's a 25% chance that you'll get an affected foal. And we'll go through that a little bit more. But so then the question for me was, well, how do you find a genetic mutation? So in a lot of horse diseases, we look to the human route. We look to dogs and cats and see what other diseases are there where they have genetic mutations, and let's just look at that gene in the horse. That's how the mutation for HYPP in quarter horses was discovered. Many of the diseases, there was a similar disease in humans. Humans don't stand on their fingernails, so there actually are genetic diseases in humans where they cause nail dysplasias, so their nails are very weak, but you never really see severe clinical signs, and nobody invests a lot of money into nail diseases in humans, right? Whereas ponies are walking around effectively on their nails. So it's a very different setup, and there is no candidate gene in humans to look at. So instead we had to take this genome-wide search. And to break it down into kind of how much work it entails, we're starting with the whole genome. So horses have 31 autosomes, which are the non-sex chromosomes, and then an X and a Y. So 33 chromosomes total. That's 2.7 gigabases. So a base, remember DNA is made up of two strands. You've got A, C, T, and G. Those are your nucleotides. A base is every one of those A, C, T, or Gs. So there's 2.7 gigabases that we need to look through to try to find one that is the mutation. So it's truly the needle in the haystack search. So what we do is we try to get from the genome down to a chromosome, down to a region of a chromosome, which will be about 2 million bases. So we're making some progress. If you can get down to a gene, then that's about 1,000 bases. So you're getting closer, and then the whole idea is to get to that mutation, which is one base. So it is not, I mean, it's kind of one of those things when I went into it, I said, oh, you just go find a genetic mutation. You know, and it sounds easy, but you're actually really trying to break it down from a huge pile of nucleotides to one. And so these are the steps that we took. So one of the principles you really need to understand in how we did this is that areas of the chromosome get inherited together. Okay, so there is crossover, there can be recombination, but for the most part, areas, hello, hi, welcome. Areas are inherited together. That's okay. We all know. So this is just a schematic of a chromosome. So each of these colors is just kind of an arbitrary piece of the chromosomes. And what's going to happen most likely is that this will get inherited together, and this piece might get inherited together, and this piece. And so we use this technology, and we actually put markers on the chromosomes so we can follow them down. So just going back to the mode of inheritance again, this is kind of what we saw with that filly that Megan had. So this is the stallion that isn't really her sire, but is a gray stallion. He is a carrier, okay? So this is a normal copy of the gene, and this is a mutated copy of the gene. And then the dam is the same thing. Normal copy, mutated copy. So all you have to do is draw what's called a Punnett square to figure out the chances that the foal is going to be affected. So there's a 25% chance the foal will be a non-carrier, totally healthy, no issues. 50% chance the foal will be totally healthy, but will be a carrier. So it will be completely normal, but could potentially pass this on and spread to another carrier. And then a 25% chance that the foal is affected, which in this case, this filly was unlucky enough to get that 25% chance. So that's the way when you're kind of looking at this, how you break it down. So we went ahead and we used these markers to do what's called a genome-wide association. And so, remember I said every nucleotide you have an A, C, or T, or G? So in this case, say I have a G here, and it's double-stranded, so its partner is a C, and yet somebody else in this room could have an A, and its partner would be a T, at the same exact region. It doesn't have to cause a disease, but it causes what makes us different. That's what makes us all different, okay? You've all got the same amount of DNA, but there's these changes in there, so we can use those as markers. So the horses have what's called a SNP chip, or a SNP array, and there are 76,000 of these SNPs spaced throughout the entire genome, so along all those chromosomes, and we use those as markers. And we say that if all the affected ponies are AT at one marker, and all the unaffected are CG, the mutation might be by that marker. And that's how we start to get down to the region of the mutation. So this would be the example of that, whereas if the mutation was by marker C, then we would actually be able to follow it down, and every affected pony would have certain allele at C, and the unaffecteds would not. So these are the results of the genome-wide association study. So we used 16 affected ponies, 17 controls. This graph is called a Manhattan plot, and it's called Manhattan because usually it looks like the skyline of Manhattan, but this looks like the skyline of Dubai. which is exciting for geneticists. So each of these is a chromosome. Okay, so that's chromosome 1, goes all the way, X chromosomes here, which is 32. There are no markers on the Y chromosome. Sorry, gentlemen, but it's not important enough of a chromosome to document any markers. There's not a lot of information on there. And then here's a negative log P. So each of these dots is a SNP. Okay, so the higher the SNP, the more significantly associated it is. So when you have something like this, I mean, it's beautiful. It's very clear that there's a region that's on chromosome 8 that is associated with this phenotype. We run a series of tests because we're doing multiple testing to cut out some of the background noise, and this red line now denotes statistical significance. So any SNP over that line is statistically significantly associated with this disease. So now we've got a region. So now we've taken it down to a region on chromosome 8. And so this was a very exciting first step as we've gone from whole genome now to a region. So when you go in and you actually look at this region, this is actually a schematic. This is actually from my Excel spreadsheet of what this region looks like. So this side is all the affected ponies. This side are all the unaffected ponies. Each color is color-coded what their genotype is. So are they CC, TT, CT, whatever color they are. Okay, so this whole region here is actually a block, and we call it a homozygous block. So every affected pony has the same genotype at that region. So if you were to look at it, they would be GG here, CC here, TT here, whereas the unaffecteds can have different genotypes. And so this narrowed this region down to 1.8 million bases. So these are markers. This isn't the mutation yet. These are just markers in this region. So now we're getting more and more closer to that one mutation. So now what do? So we've got a region. We know it's on chromosome 8. We know it's in this region. The next step is to sequence. And what you can do is you can actually go and just sequence this region. And that's what we would have done probably even only 2 or 3 years ago. We would have just gone and sequenced that region because it's cheaper. Nowadays, it's cheaper to just sequence the whole horse, or the whole one, which is amazing. So remember, 2.7 gigabases, we're sequencing the entire genome, and then we're just going to look at that region, because that's the region we care about. So we did 2 affecteds, 2 controls. It's still pretty expensive to do the sequencing, but we started with that, and that was enough. And we got coverage. When it says 6x coverage, that means that there's 6 copies at every region. So it just kind of tells you depth. And so what we're looking for are SNPs, which remember was are you AC or TG? And then the other thing we look for are insertions or deletions. And so in this case, this would be normal. This sequence would be a completely normal animal. This sequence would be like if you were typing and you inserted a T by accident. So that's an insertion. That can cause disease. Or the opposite would be if you were typing and you forgot to put the G in. So now you've got a deletion. So we call those indels. So this is what we're looking for, SNPs and indels. And so this is just a schematic of what this looks like. To give you guys an idea, each of these gray bars here is a read. So we chop the DNA up into a bunch of pieces, and then we sequence all those pieces. And then you have to reassemble all those pieces. So we have a reference genome. We have a horse that we already have sequenced. We know where all the genes are. And so we line them back up. So here we're just lining these all back up to the reference. And then each of these colors here is where there's something different, meaning that this particular, whatever it is, A, C, T, or G, doesn't match up to the reference horse, which is thoroughbred. And that we need to look at that area a little bit deeper. So this is one pony and just an example of that. And then when we go in, we can find all of these variations. So here's all the 4 ponies lined up. So this was a SNP. You can kind of look down and see the colored reads there. This was an insertion or a deletion. That's the black dot. There was another SNP here and another SNP here. So we go through that whole region and we look for things that are different. And we pull those all up. So what we ended up getting was in that region, there are about 9,700 SNPs and 1,200 indels. So these are all possible mutations. But then we looked and we said, well, what's different, affected and control? That's what we really care about, right? Where the affected are one thing and the control are another. And we got it down to 363 and 28. And then if we just say, well, what about within genes? There were 17 SNPs and 5 indels. So we've got 22 possible mutations. So we're down from 2.7 gigabases to 22. Which is exciting and in a really good spot. So the next thing we did was we designed a custom array, and we used 369 Connemara ponies, 23 are affected. We also used non-Connemara ponies, Arabians, quarter horses, and thoroughbreds. This disease, to date, the the way we have classified it phenotypically has not been observed in these other breeds. So we're going on the premise that whatever the mutation is, we should not see it in these breeds, and we didn't. Only 3 of those 22 mutations segregate in 3 genes, so now we're down to 3. Now we've got 3 possible mutations, and they're in 3 different genes. So we've kind of gotten down here, we're at 3 different mutations, and then the next thing you can do, you can't really map anymore, you've kind of gotten as down as you can. So one of the next things you can do is look at expression. And the reason why your eyeball is different from your finger, is different from your leg, is because not every gene is expressed in every tissue. So certain genes are turned on, certain genes are turned off. So we were really interested, well, which of these 3 genes is expressed in the coronary band, right? We tried to do dorsal hoof wall, and it is incredibly difficult to grind that up and actually make RNA out of it. So we had samples from the coronary band. So this was a very cool day. So there are 4 genes on here that were candidates. Okay, these 4 genes. Beta-actin is a housekeeping gene. This means it's expressed in every single tissue. So we put that just to show that it works in every tissue. And then you have all different tissues here. Spleen, heart, lung, kidney, stomach, liver, skin, coronary band, brain, spinal cord, and then negative control. And so, hang on. We've got here 3 of the 4 genes were expressed in coronary band, but 1 was very, very strongly expressed. So the brighter the band, the more expression you have, especially when your control looks as good as it does here. And so when we went and actually looked at affecteds versus controls for this gene, so this expression is on the y-axis, the control has 100% expression, whereas the affected is down at about 10%. So there's definitely a difference in expression in the coronary band of affected ponies with this disease on this gene. So now we've actually gotten it down to 1 base. And I'm not providing the gene name because the paper is about to be submitted, and until it is submitted and actually accepted, we will not be disclosing the gene name. However, we did get the patent to go through at UC Davis, and so we've actually have the test available, and it will be up and running in a month. So to date, we've tested 31 ponies that have tested positive, and I can update this now. So we had 1 pony that was 3 years old that had no discernible abnormalities, and these were as determined by us by digital photographs. So we contacted the owner and said, can you please send us pictures of this pony's hoofs? They tested positive, but it doesn't sound like it's positive. And the owner has graciously agreed to let me say that when we look at actually the solar margin, we looked at some pictures today, it does appear to be mildly affected. So it's 100% penetrant, meaning that if the pony's got the mutation, it's got the disease. That being said, there's a spectrum. Okay, not every pony is going to be as severe as those pictures I showed you. There's a lot that are mildly affected. But the mutation is the mutation, and if it's fully penetrant, which most autosomal recessive diseases are, it's just going to be on that spectrum of mild, moderate, severe. So I'm going to skip over the whole incomplete penetrance thing because we don't need to talk about that. But I had poker stuff for any poker fans in here as an analogy, but talk to me about that later. So as I said, we're going to have the test available. It should be up and running in a month. We just ordered the primers, and then we just need to run the controls and make sure everything is good. The cost is $40, so it's $40 US, which is really reasonable for a genetic test. And it will be, all you have to do is go to Google and type in UC Davis VGL, which is the Veterinary Genetics Laboratory, and this page will come up and you'll find the links on there. It's really easy to access. Hair samples are what needs to be submitted for testing. So hair samples with roots, they usually say 30 or 40 out of pull from the main and test. And these are the results you'll get back. Okay, if the pony's NM, there's no copies, the animal's normal. So the pony is NHWD, it has 1 copy, so it's normal, but it's a carrier. And then if it's got 2 copies, it's affected. As of right now, the carrier rate that we've detected on our research samples is 15%, which is pretty high. A lot of them, if you look kind of across the board, a lot of autosomal recessive diseases are somewhere in the 8 to 13% range as far as carrier rate. And this is a biased sample set, right? Because these are samples that are only sent for research. So as we get more and more samples, we'll be able to update that carrier rate. So to conclude this, this is actually an inherited hoof wall defect in Connemara ponies that we've seen so far. Affected ponies can be managed, but are often have a tough time performing. And then again, the mode of inheritance is autosomal recessive, and we can use genetic testing. And so I just want to address one really important fact. This is an autosomal recessive disease, and because of that, you guys are actually incredibly fortunate because the carriers are completely healthy. So yes, they carry a copy of this particular mutation, but most of these ponies, they may be actually very high performance ponies, and there's no reason that they shouldn't perform and be bred. I think that genetics sometimes gets a bad name, and you think that any animal that's a carrier for something should never be bred, and that's not the case. There's a lot of traits you like about these ponies, which is why they should be bred. However, you just want to make sure you don't breed them with another carrier. And so in this case, if you had a sire here that was a carrier for this mutation, and you screened a bunch of dams, and you found one that was not a carrier, you've got a 50% chance of a non-carrier and a 50% chance of a carrier. And as long as that offspring is tested if it's going to be bred the same way, you can still breed these animals. And that's the smart way to stay away from this disease and yet still keep the phenotypic and all the qualities you really like about these ponies. So I have a very strong proponent. This is not like HYPP. So HYPP in the quarter horses, for anyone that's familiar with it, is an autosomal dominant disease. If you have one copy, you have the disease. There's no carrier status. This is recessive. Yes?
In the non-Connemara controls at 150, for any of those carriers?
No, not a one. And we have not detected it in another breed yet, and that's how many we've tested so far.
So you the non-carriers, in the other breeds 150, which you do not have a wider.
Right, we did not. And so what we're going to be doing now is hopefully, and all we had were quarter horses thoroughbreds already, and so it'd be nice now to get other breeds and to see if we can find carriers among other breeds, because it may very well be the case.
Like I presume you're calling it Hoof Wall syndrome, you're not calling it Connemara Hoof Wall syndrome.
Nope, we're calling it Hoof Wall Separation Disease, and we're not calling it Connemara because even though that's the first breed identified, it's just going to be like a lot of other diseases where you start to find it in other breeds. You are correct. So this is really kind of a strong point for me, is that I hate to be the bad guy geneticist all the time, and I really think that it's great to have a test, because now you just breed smartly. And you just don't breed two carriers, and you'll never get an infected foal, and yet you can still keep what you like about particular carrier ponies. And we're fortunate because of this. If it was dominant, we couldn't do this. So having the test is going to be a huge, huge asset. So thank you again to the co-authors, all of the owners of these affected ponies. I've heard so many stories, and a lot of these are pictures that owners sent me. That's kind of what they've been through. The referring veterinarians and farriers, and special thanks to Jocelyn and Penny, who really kind of pushed this forward. And it's an amazing example of what a community can do when they're really passionate about something, and they just keep hassling me to get it done. And they did, and I did. But I think that they deserve a round of applause for everything they've done. Take any questions. Yes?
If you test, how many generations would it take to get the disease out of the breed?
Well, it depends on what you do with your carriers, right? If you decided, well, we're never going to breed a carrier again, you could get rid of it pretty fast. But I don't think that that's going to be...
All individuals included. If you all want to breed with all the individuals,
right?
How many time would it take if you test?
If you did the testing, so you want to get rid of it completely, you don't want any carriers. Yes. So you're probably looking at 7 to 10 generations if you were really smart about it. But I just have such a hard time. There's so many good things about the carriers, you know what I mean? So I think that's...
I don't want to exclude ponies, right. I just want to know...
right, what it would take.
What? And
I will tell you that, so quarter horses, and many of you may know, have more genetic mutations uncovered than any other breed. And it is not because they're unhealthy, it's because the American Quarter Horse Foundation funded genetics research, whereas many other places didn't. And so now they have a 5-test panel on their, so AQHA, the American Quarter Horse Association. Every quarter horse that is to be a breeding animal must be tested for all of those diseases, and decisions made based upon that. And they really had a hard time. When it first came out, when HYPP first came out, AQHA got a lot of pushback that people didn't want to test, but it's become where people that are buying these horses want to know, and people that are breeding horses want to know. And it's for the health of the breed. It's really kind of made a big difference, and it's been a big shift. But I think it's just something, to have a test is huge. It really is huge. Yes?
Is this something that's just evolved recently, or has this been in the breed for a long time?
So Sheila's going to talk a little bit about that, and I can't answer that unless you gave me all the samples from years ago to test, and I wouldn't answer that unless you gave me all the samples. I am a very strong believer in if you have a genetic test, then that's the way you qualify that. You don't look at pictures from 20 years ago and say, oh, it looks like it could have been. But Sheila's going to talk a little bit about the pedigrees that she was able to follow back and her thought on it. But again, unless you have samples from those, it's kind of like impressive with the quarter horse disease. They didn't go further back because they didn't have samples. So the buck stopped with him, and that's the way it should be.
Thanks.
Yes?
It's to test that any lab doesn't want to really have to send samples to you.
So we filed a patent,
Okay.
which means that UC Davis has the license. However, genetics labs can apply for the license. So they pay a fee. They get, a yearly, annually renewable license while it's still on patent, and then they can run it. But then they run it with our primers, our controls, yes. And that will be what Davis ends up doing. They will license it out. That's what we.
Once you microchip in your phone, you can send a sample out. When you get the sample back, you know if you're a carrier.
Absolutely. Yeah. And the turnaround time, they're really quick. I mean, they're usually, I think, 3 to 4 weeks is their turnaround time. And they will send you the piece of paper with kind of the breakdown and the carrier rate on it. Yes.
No, it's just that on the internet, if you Google it, it can come up Connemara Hoof Wall syndrome.
Right.
You're said, as you're sending in your paper.
We're going to, yes.
It's going to say don't on Connemara.
It was discovered on Connemara.
Yeah, yes.
Right.
But the thing would be misleading by saying Connemara Hoof Wall syndrome.
Well, right now, we're calling it Hoof Wall Separation Disease, and then we do have that we've identified it in Connemara ponies.
Yeah.
So we are stipulating that. Until we find it in another breed, I will
No,
be...
but I'm just saying the internet starts with Connemara.
Right. I understand.
That's just how the search engines work.
Right.
Because the publicity has been to do with Connemara, so the search engines
It's going to pull that up.
Yes,
That's just, I think, the society here, the parent society, wouldn't like to see that being published in that manner.
Well, and we will, and if we find it in another breed, it will really push that forward. But for right now, I think hoof wall separation disease is an appropriate, it's not a syndrome anymore, we have a mutation, it's a disease. So I'm happy calling it that, and just abbreviating HWSD without Connemara in there, but the research has been done, of course, in the Connemara breed. Yes?
I was just wondering, your original sample size for step 1 was quite small.
It was amazingly small.
Yeah, so I was just wondering, how do we know it's not like a sire effect or grand-sire effect?
So we actually calculated genomic inflation on that population. So we looked to see if there's any cryptic relatedness in there. And it was, genomic inflation was 1. So anything around 1 is considered, no chance of a false positive. And we also didn't pick, we picked half siblings. So we had offspring, we didn't put sires and dams on the array. It was just offspring and then half siblings. So we controlled, because trust me, my other equine studies don't go this well as far as mapping. And so we really controlled for the cryptic relatedness based on that. And the other reason we were lucky is it's also more recessive. So when you do that simple chi-square, the stats go up. And it's also because it seems like there's a very long section of linkage disequilibrium. So big chunks of the chromosome are inherited the way the ponies have been bred. So we were lucky because of that. Yes?
What are the first symptoms that a pony do have it? Or are they, do they have it before you notice it? Do you know what I mean?
So, sorry, say it
The
again?
first symptoms now that a pony do have it,
Right.
what do they show?
If a pony is affected?
Yes, yes.
So typically that dorsal, it looks like white line disease. To most vets that I've talked to, most farriers, they get that kind of retraction of the dorsal hoof wall. But anything that you're suspect of now, you don't really need to worry that much. I mean, send the genetic test in. That's going to be the...
Yes, but doesn't it look a lot like that it's an effect from laminitis?
No.
So, so the lamina are actually completely normal. The ones
When
we
you see
looked at.
like a laminitis in a pony, the after effects, the hooves can go that way.
Right,
Yeah.
they can.
And
But
like,
these ponies aren't founders. When they initially present with the
Isn't it
signs.
possible that with laminitis, the hooves get as bad as ponies have to be put down?
Right.
Yeah.
Well, that's the case,
So-
but in these, laminitis is secondary.
You know,
It
like,
follows this.
you see what you've said there, like, I don't know, but it's just back years ago. I remember when I was young, and that's not today or yesterday. Like, I did see horses that had something like that,
Right.
but the old people said that it came from laminitis.
chicken or the egg,
Huh?
you know? I mean, in our cases, we've seen them now where this comes first and they are
Yeah,
not
you see,
rotated, they are
it's
not
probably
foundered.
and it, like nearly all horses in at that stage.
Right,
There were horses that were fat, like that was overweight,
right,
you know, that caused, and which do cause laminitis anyway.
right. Yeah,
I don't
laminitis
know,
can be
it
a
looks
sequela
awful
to
like it, you
this,
see.
but but laminitis doesn't cause what we're seeing here as far as this particular mutation. This happens way before any laminitis down the road. And laminitis, as you know, is incredibly complicated. You've got obesity, inflammation, insulin, you've got all these other factors that play a role. And so on this, the inflammation of that hoof, I think, sends them into that stage where they may founder. They don't all founder, but they may founder. So yes, it's something you can see. And again, if you've got something in the field and you're not quite sure which came first and it's already foundered, yet its hoof wall's cracking, run the genetic test and see. You know, at least you're going to know, did you have that underlying?
Yeah, we actually had a pony recently, an old pony, like 18, and when the gal bought it, she was told he had laminitis, and he looked like he had a laminated hoof. But with a lot of good shoe and stuff, it actually was looking pretty good. And when she sold him fairly recently, and he went through a full pre-purchase, and there was no indication of laminitis whatsoever. He was a Connemara, but unfortunately without the hadn't passed the back, so we didn't have a breeding with him. But that was, they thought for years he was laminitic and he wasn't.
Well,
And he was not ever on sound actually.
Right. Remember, laminitis is a spectrum too, right? You can have ones that don't rotate and ones that don't sink.
But there was, yeah.
Yeah. So now you've got a test. Now you can do it. Makes life easier for vets. Yes.
It's that they're like to compliment you highly. It's absolutely amazing.
Thank you, sir. Thank you very much for that.
I was just wondering how many countries sent blood samples in for the testing initially.
Sheila, how many countries sent samples in for the testing? You would know the answer better than I.
Yes, for the initial work, the very beginning of the work, there was 18 ponies in 6 countries. There was 2 add-ons to your original 16. That's 18 ponies from 6 different countries.
And all of the owners paid to ship those samples in on their own dime. I have to acknowledge Morris Animal Foundation paid for the genome-wide genotyping, but then the rest of this has been paid for by owners and by donations, which is, again, a phenomenal feat and a real testament to your organization. Yes?
And if I remember correctly, gene C was also expressed in the stomach.
As they all were.
They all were. So is it because the animal is not able to digest or absorb nutrients correctly? Is there a treatment that's available?
No, it's once the gene comes out, it gets to be a little more, it makes some biologic sense as to why that is, but it looks like it's maybe part of an enzyme that would also be secreted either in the pancreas and then get into the stomach, or there's that family of genes seems like there's some enzymatic function. So it makes biologic sense since we kind of are able to disclose the gene. But yeah, that was a really interesting finding that they were all expressed and that was highly expressed in the stomach. Yes?
Would it make sense that natural selection in the past was more common than today? In other words, it was always there, but you might have been up in hamburgers before now. Now that people are more sympathetic to them, they keep them and they breed them and they sell them on to other people. So
it could be.
Is that the difference between 40 years ago and now?
Right, it could be. I mean, the other thing could be that, like a lot of the equine diseases, you know, I brought up HYPP a lot, that got perpetuated because in the show ring they liked the well-muscled horses, right? So that kept going because those horses were winning and so they got bred. And these horses, at least from what I understand, I'm not a Connemara breeder, but the carriers can be quite typey and very sport horse looking, which is really kind of gotten to be a big niche for the Connemara breed. And so I wonder if that shift a little bit into the more sport horse looking, you know, they really like the way they look, they like the way they go, has kind of perpetuated this. I don't have any proof of that, that's just a theory. But
Natural selection in the past would be that it work, they wouldn't live. Nowadays, things can live and they don't have to work at all.
you're exactly right. If you had feet like this and you were pulling a carriage out here, you would not have made it.
Yeah.
For
It's
sure.
quite possible that that's
Yes. Yes.
Just on the recessive, like with E, I think it's 25%, but your percentage was lower, but you actually got.
For the carrier rate, you mean?
Yeah.
So that was just testing a random, not even random, that was testing a biased population of samples that got sent to us. So all of those 269 Connemara's that we had samples sent that people said they look normal. We tested them and 15% of them were carriers. So it's going to be lower than the 25% because it's not, we don't have 100% of all the Connemara's that were ever bred and were ever assessed.
We got another sample, it could be lower. Or
It could be lower or higher, yes. It's a completely biased.
Yeah.
So that's what I look forward to when we have bigger numbers, when we're in the thousands, you know, and looking at carrier rate, that would be much more accurate than that rate.
And the other equines, then, you want to widen your sample based on other equines.
Yes, and we're actually going to try to recruit some other breeds that may have kind of some similar looking disease and test them for the same mutation and see if they have it. That would be really the next step. And if they don't, then they need to fund their own project. But if they do, then they've got to test too.
You said you tested 150 or so non-Connemara's and not found a single carrier, correct?
Correct. So other pony breeds, Arabians, thoroughbreds, and quarter horses. But we haven't done all the other breeds there are, right? The illusions of that.
Did it affect the half breeds, Connemara?
We have not, so we have not seen one yet, have we? A half-breed affected. They can be carriers.
Oh, they can be.
But you would have to take, right, you would have to take a half-bred like a Connemara thoroughbred that was a carrier and a Connemara thoroughbred that was a carrier and breed them together to get a chance of it. So we have not seen that yet. But it has the potential if you had half-breds that were carriers, yes. So we have had those be carriers.
Carrie, if you go to other breeds, as you probably know, the Connemara is a convergence of other breeds. So the quarter horse most likely will never match us.
Exactly.
But there are 5 other opportunities.
There are, and I'm eager to get samples from them. All your friends.
Yeah.
Yes.
So sorry, who's going to fund the next part of your research?
Well, the good thing is that they're really, once the test is there, owners submitting samples, so every sample that gets submitted to the lab is barcoded, confidential, and the results are sent to the owner. However, for research purposes, we have access to those samples, so I can go in a year from now and say how many of this breed, without the owner's names, but just how many of this breed tested positive, how many of this breed were carriers, and so that actually doesn't cost a thing. Which is quite nice. So the expensive part of the project is done, which I never get to say. I'm always asking for money. So yeah, the test is out there, and that is just going to be now accumulating data based on the test results. And that I think will really help owners of other breeds, you know, as well as the Connemara breed, kind of make decisions. But the test is there. It'll take care of itself.
It's terrific.
All right.
Can you tell Peter how much the research at the Morris Foundation?
So this whole experiment cost us $170,000, and Morris Animal Foundation put in about $110,000, and then a lot of the rest of it came off of our individual grants, and then Connemara owners and breeders that donated money made up, especially the sequencing. They really paid for the sequencing. So it was a very, very big joint effort, and we are incredibly grateful to everybody. Again, kept pushing both. You know, intellectually and financially, that was a good thing. Okay, thank you all for your time.
Thank you.